Good morning, student. Today we are going to discuss about chapter 9, biomolecules. Here you see the textbook part. The biomolecules is a compound, that is the either elements or compound found in the living organisms are called biomolecules. So biomolecules actually deals with the different organic and inorganic compound, and I will follow the follow another book that is called Companion Biology. Here you see the chemical compositions of biomolecules, as you see in case of elements found in earth crust as well as in human body. Here you see the elements are hydrogen, carbon, oxygen, nitrogen, sulfur, sulfur, sodium, calcium, magnesium and silicon. These are found in earth crust as well as in human body also. And by chemical analysis we can detect the compound present in the human body. Here, besides the organic compounds, we found that there are some inorganic compounds found in the human body or biological organisms. There is sodium, potassium, calcium, magnesium, water, and compounds such as sodium chloride, calcium chloride, phosphate, and sulfate, etc. So we see that after getting the S that contain inorganic compounds that I have already mentioned here, calcium, magnesium, inorganic compounds like sulfate, phosphate, etc. And organic compounds are also found. Those are, we can say, amino acid, nucleotide, fatty acid, lipid, etc. So in this chapter we first discuss about the definition. So what is biomolecules? All carbon compounds or molecule present in the living tissue are called biomolecules. So biomolecules are actually the carbon compounds, and the carbon constituents are more than 50% of the dry weight, and that is called found in living tissue are called biomolecules. These biomolecules are essential organic compounds as well as inorganic compounds. Essential organic compounds are called metabolites, and there may be primary metabolites and secondary metabolites. That means there are two types of metabolites. One is primary, other is secondary metabolites. The primary metabolite, here you see the primary metabolites actually are pigments, alkaloid, essential oil, toxin. These are secondary metabolites. These are pigments, alkaloid, terpenoid, essential oil, toxin, lecithin, drugs, polymeric substances. These are secondary metabolites. And what about primary metabolites? Primary metabolites are basic metabolites, or they are found in living organisms. Primary metabolites are intermediate products of normal vital metabolic pathway of organisms, such as in respiration, digestion, assimilation, etc. These are amino acids, these are nucleotides, sugar, fats, fatty acids, peptide, etc. So biomolecules and about primary and secondary metabolites we have already discussed, and the composition, average composition of cells, water found in cell, that is 70 to 90%, protein, that is 10 to 15%, carbohydrate 3%, lipid 2%, nucleic acid 5 to 7%, and ions 1%. So by chemical analysis we can get the average composition of cells of different components in a percentage manners. The biomolecules are two types. They are micromolecules or macromolecules. Here you see the difference between micro and macromolecules. The micromolecules, they are which are soluble in trichloroacetic acid. That means the molecules, those are soluble in trichloroacetic acids, are called micromolecules, while macromolecules, they are not soluble in trichloroacetic acid. Secondly, the molecular mass in case of biomicromolecules is up to 800 Dalton, while in case of macromolecules, macro means large, macromolecules, it is above 10,000 and above. Biomicromolecules, micro means minute or small molecules, they are monomers, they are called monomers, while macromolecules, they are generally called polymers. So biomolecules are two types, one is micromolecule and other is macromolecules. In the micromolecules, we found the inorganic salts such as the list here shows that sodium, potassium, calcium, magnesium, water compound. These are inorganic salts, and the linking, the nature of bonds and linking monomer in a polymer, how they link together, how the micromolecule converted into macromolecule in living organisms by the help of a certain bonds. There are three types of bond found in macromolecule: peptide bond, glycosidic bond and phosphodiester bond. Peptide bond, it is called amide bond. Here a peptide bond is formed by reacting between an amino group, that is NH2 group of one amino acid, with carboxyl group of COOH group of, that is called carboxyl group of another amino acid, with the loss of one molecule of water. So in the formation of peptide bond, one molecule of water is released, and how the bond is formed between the two amino acid. The first amino acid amide group react with the carboxyl group of second amino acid, and the bond is a covalent bond, and it is CONH peptide bond. It is found during the synthesis of protein, that is formation of dipeptide, oligopeptide, polypeptide, etc. Second bond is glycosidic bond. It is found in formation of polymers of carbohydrate or polysaccharides or oligosaccharides. That means it is found in carbohydrate and carbohydrate containing complex compounds. Here the bond occur between carbon atom of adjacent monomers through any type of group, such as either carboxylic group or amino group or amide group, etc., which can provide hydroxyl and hydrogen for synthesis of water. So in this bond also, it occurs between carbon oxygen carbon or between carbon nitrogen carbon, but here one molecule of water is released. It is called glycosidic bond. Third bond is phosphodiester bond. Two ester bonds are established between one phosphate radical and two adjacent pentose sugar of nucleotide. So it is found in the formation of DNA and RNA nucleotides. Those are the macromolecules of DNA and RNA, and here a molecule of water is released. It is called phosphodiester bond. O dash phosphoric acid dash O, that is oxygen phosphoric acid O. That means two carbon pentose sugar react with phosphoric acid, and in this way nucleotides are linked with one another during the formation of a polynucleotide chain. First, we discuss about carbohydrate. What is carbohydrate? Carbohydrate is actually a polyhydroxyaldehyde or ketone and their condensation product. That means it may contain aldehyde CHO group in terminal or keto group in the second carbon atom, and it is and it either form the aldose carbohydrate or ketose carbohydrate. That means in carbohydrate we found aldehyde or ketone, that is called polyhydroxyaldehyde or ketone group. So if it contain aldehyde, then it is, these are called aldose, and if it contain keto group, then it is called ketose. The term carbohydrate is generally derived from French word hydrate, decarbony or hydrate of carbon, meaning thereby that carbohydrate contain along with carbon, hydrogen, oxygen in proportion as present in water. The empirical formula is CnH2O whole n or CH2O whole n, where the value of integer n is 3 to 7. That means n defines or denotes either 3 carbon or up to 7 carbon for monomers. Carbohydrates are also called saccharides. Saccharides mean sugar, as they are formed of sugar. The term sugar is used for small carbohydrate, that is monosaccharide and disaccharide. Those are sweet in taste. Carbohydrates are the first product of photosynthesis and the main ingredients of reserve food. They are also called stuff of life. Up to 80% of dry weight of a plant is made up of carbohydrate. However, in animal, the carbohydrate content is not more than 1% of total carbohydrate and are of 3 types. So, carbohydrate are of 3 types: monosaccharide, oligosaccharide and polysaccharide. So, in our next class, we will discuss about the difference between monosaccharide, oligosaccharide and polysaccharide.